Hello, my name is Jocelyn Hach, and today I will be doing my local expert presentation on the Western blot. So what is a Western blot? A Western blot is a technique used in laboratories to detect proteins in a sample that uses the size of the protein and the protein-specific antibodies linked with a reporter enzyme to determine the presence of a specific protein. Westerns are often used as validation that you have the correct protein for research articles and papers. Westerns also allow for the visualization and quantitation of the target protein. So here's a quick overview of the Western blot workflow. First, you need to do the sample preparation, and then the gel electrophoresis, which you can see here. And then you transfer, then you block, and then you put on the primary antibody, wash it, then do a secondary antibody addition, and then from there you do the detection and then the analysis. So let's start first with the sample preparation. The cells are lysed to release cellular proteins using lysis buffer or other forms of homogenization. The protein is quantitated to make sure there will not be too much or too little protein when the gel is loaded. So here is an example of a protein quantitation using BSA. Gel electrophoresis. So the proteins are treated with a reducing agent to unlink the disulfide bonds using either DTT, dithiothriotol, or BME, beta-mercaptoethanol. They're then put in an SDS page buffer to denature the proteins along with a loading dye to visualize the sample. The sample is loaded onto a polyacrylamide gel along with a molecular weight marker and run with the SDS buffer. An electrical current is applied to the gel, and the proteins with lower molecular weight move faster through the gel, while the larger ones move slower. Next, we need to transfer the proteins to our membrane. So after the electrophoresis, a membrane, usually PVDF or nitrocellulose, is placed on a gel along with filter paper and transferred via electrophoresis. Usually you can see here there is a type of sandwich made, including filter paper, the membrane, and the gel. Here you can see the most commonly used, or at least in my day, commonly used transfer apparatus by Biorad. There's also this semi-dry transfer, which I use quite a bit as well. Next, we have the blocking of the membrane. So once the proteins have finished transferring to the membrane must be blocked to prevent nonspecific binding of antibodies as well as to reduce the background signal. Blocking solutions should contain proteins like BSA or dehydrated milk. Blocking should be done long enough to be effective. Usually overnight is sufficient. Next, after the blocking, we add the primary antibody. So after blocking, the membrane is incubated with a primary antibody that is specific to the target protein. The proteins have been tagged with a marker that is detectable by the antibody, such as an HA or His tag, which would be an anti-HA or an anti-His. And this would be used in that case. You must know which animal the antibody was made from in order to determine what the secondary antibody is going to be. The antibody concentrations vary depending on the manufacturer. The manufacturer usually wants you to use more than is necessary, so it is a good idea to test the appropriate concentration first so none of it is wasted. They can be very expensive. Next, we need to wash off the primary antibody. The membrane containing the primary antibody must be washed off to prevent a high background signal. Wash buffer usually contains TBS, tris buffered saline, or phosphate buffer, saline, PBS, and 0.1% tween 20. The number of washes can depend on the protocol and the antibody. Next, we have the secondary antibody incubation. After the membrane is washed, the secondary antibody is added. The secondary antibody will bind to the primary antibody and will likely contain a fluorophore or chemiluminescent property for detection. The secondary antibody must be made in an animal that the primary antibody was not made in for it to bind. If the primary was made in mouse, the secondary must be another animal, goat for instance, making it goat anti-mouse. Here you can see a nice description of that, and then the two antibodies binding together. So detection. There are several types of detection methods that are used in Western blotting to visualize and quantify the proteins of interest. The choice of detection method depends on factors such as sensitivity, equipment availability, and the specific requirements of the experiment. Here are two common examples. The first will be chemiluminescence. So chemiluminescent substrates generate light when they react with an enzyme conjugated to the secondary antibody. They're highly sensitive, have a wide dynamic range, and they're compatible with film or digital imaging systems. Enhanced chemiluminescence is a widely used chemiluminescent detection system. Next, we have fluorescence. Fluorescent dyes emit light when excited by a specific wavelength. Secondary antibodies are often conjugated with fluorophores. Multiplexing capability, which is detecting multiple proteins simultaneously, is very advantageous, and there is no need for film development. Some common dyes would be Alexafluor, Sci-dye, IR dye, and there are many others as well. Here are some example detectors. Here is a chemiluminescent detector, the ChemiDoc by Biorad. Here is an Invitrogen iBright FL1500, which can do, I think, both chemiluminescence and fluorescence. And here is the old school film developer, which is pretty much obsolete, but this is what I used to use when I was doing a lot of Western blots. So results. Once the membrane has been imaged, the protein bands are analyzed in many ways. First, you can do protein size. The size of the protein is determined by the molecular weight marker, or ladder, on the membrane. The size can also be indicative of a post-translational modification or change in protein size due to dimerizing or many other types of additions or subtractions to the protein. Another form of analysis is done by densitometry. So this can help you determine the amount of protein present. So the amount of protein can be determined by the pixel intensity within a defined region. Quantified standards need to be present in order to determine the amount. And some of the imaging software that I used in the past was ImageJ. You can also determine presence absence, which is a very obvious way to tell if your protein is being expressed or not. And these are just some of many types of things you can do. So here is an example of an experiment I did many, many years ago. And you can see here, this was done on film. You can see my this is hemagglutinin. And and then you can see an extra thing you can do is test with Kumasi stain to see, visualize the amount of protein in your, to make sure everything is even. And with that, thank you very much.